Good afternoon, everyone. Welcome to the Biology School Complex session. I will start the session with an interesting question. A couple comes to you as you are their trusted biology teacher with a question about their pregnancy. They have a dominant genetic disorder that runs in the husband's family. The wife is now pregnant. They also mention that the wife has a history of an autoimmune disorder which could potentially affect the placenta during gestation. Now, the questions to answer are: predict the baby's genetic risk according to Mendel's laws, ensure the successful development of the embryo and fetus. I know that you all will definitely answer these questions. Baby's genetic risk is explained by Mendel's law of segregation. We must establish the genotypes of parents, specifically the husband. Since the disorder is dominant and runs in the family, the husband is likely heterozygous. According to the Mendel's law of segregation, the probability of the husband passing the disease causing allele to the offspring is 50% at the moment of fertilization. Coming to the wife's problem, we must advise them to visit a doctor. The doctor must monitor the wife's autoimmune disorder and its potential impact on the placenta. The placenta's primary role is nutrient, gas, and waste exchange between the maternal and fetal blood supplies. Damage due to autoimmunity could compromise this function, leading to fetal growth restriction or distress. To answer these type of questions, everyone must be aware of the topics related to 10th class biology present in reproduction and heredity chapters. The topics we are going to discuss in this session are: the difference between the fetus and embryo, extra embryonic layers that surrounds the fetus, how placenta is useful for nourishment, the relationship between the placenta and the umbilical cord, and a model lesson plan relates to Mendel laws. Fertilization is the fundamental process of sexual reproduction where a female gamete, egg or ovum, and a male gamete, sperm, unite to form a new cell called zygote. It is the earliest stage of development and lasting for first few days, approximately 4 days. The zygote rapidly divides through a process called mitosis as it travels down the fallopian tube towards the uterus, eventually forming a ball of cells called blastocyst. Coming to the embryo, an embryo is the term used for the developing human after the zygote or blastocyst has implanted into the uterine wall. The embryonic period lasts from the third week to the eighth week. This is the period of intense cell differentiation and organogenesis where all the major organs and the body structures like heart, brain, spinal cord, and limb buds are formed. Because all the basic body systems are establishing their foundation, the embryo is most vulnerable to damage from harmful substances called teratogens, substances causing birth defects during this stage. As we all know that embryo transforms into fetus. A fetus is the developing human inside the mother's uterus after the 8th week of fertilization until the birth. It develops from the embryo, which gradually forms all the major organs and body systems. Let us discuss about the embryonic layers that surrounds the embryo and fetus. Coming to the human embryonic layers. Human embryonic development is characterized by the formation of three primary germ layers: ectoderm, mesoderm, and endoderm. These layers play a crucial role in differentiating into various tissues and organs, laying the essential groundwork for human physiology. You are observing embryonic and extra embryonic layers in this slide. Embryonic layer ectoderm becomes hair, nails, skin, and nervous system. Mesodermal layer becomes circulatory system, lungs, skeletal system, and muscular system. Endodermal layer becomes digestive system, liver, pancreas, and lungs. The extra embryonic layers, also called fetal membranes, are four distinct structures that develop from the fertilized egg but lie outside the embryo proper. They play crucial roles in protection, nutrition, respiration, and waste disposal for the developing embryo and fetus. These four membranes in humans are amnion, chorion, yolk sac, and allantois. Let us move to amnion. Coming to the functions of the amnion. Amniotic sac creation. The amnion forms a protective sac filled with amniotic fluid which cushions the embryo and maintains a stable temperature crucial for development. It is also useful for fluid management. It regulates the production and absorption of amniotic fluid, providing a supportive environment that helps to prevent external pressure on the developing embryo. It is also useful for the movement facilitation. By allowing the embryo to move within the fluid, the amnion promotes musculoskeletal development essential for later movements after the birth. Coming to the functions of chorion. Chorion is useful for outer layer formation. The chorion serves as the outermost membrane, playing a vital role in implantation and interaction with the maternal tissue for nutrient exchange. Here you will observe the chorion layer. Its another function is placental development. It participates in the formation of the placenta, regulating nutrient and gas exchange between mother and embryo, which is critical for fetal growth and development. It is also useful for hormonal secretion. The chorion synthesizes hormones such as human chorionic gonadotropin to maintain pregnancy and support the growth of embryonic tissues. Let us discuss about another extra embryonic layer called yolk sac. Its main function is initial nutrient supply. The yolk sac provides essential nutrients to the embryo in early development before the establishment of the placenta. It supports early cell division and growth. It is useful for blood cell formation. It plays a crucial role in early hematopoiesis, producing red blood cells that transport oxygen to the embryo during a critical growth phase. It is also useful for germ cell development. The yolk sac contains primordial germ cells which migrate to the developing gonads later in gestation, essential for fertility in the future. Let us move to the fourth and last extra embryonic membrane called allantois. Coming to the functions of allantois, it is useful for waste storage. The allantois collects the waste products from the embryo, acting as a temporary storage site that helps to prevent toxicity during development. It is useful for vascular contribution. It forms blood vessels that contribute to the umbilical cord, facilitating the exchange of nutrients and gases between the mother and embryo. It is also useful for fetal urine function. The allantois facilitates the development of the urinary bladder and participates in the excretion of fetal urine, aiding in fluid balance during the development. Let us learn about the structure of human placenta. The placenta is a special temporary organ that develops during the pregnancy in mammals, including humans. It forms a connection between the mother and the developing embryo through the umbilical cord. Coming to the anatomy of the placenta has a disc like shape and contains numerous villi that increase surface area. The placenta consists of the chorionic villi, which invade the uterine wall to establish nutrient exchange. The placental layers are crucial in preventing maternal blood from mixing with fetal blood, where facilitating nutrient transfer. Here we can understand that mother's blood never mixes with the fetal blood as it transfers the nutrients through a process called diffusion. Coming to the functions of the placenta. Placenta is useful for nutrient transfer. The placenta selectively transports amino acids, glucose, and fatty acids from the maternal blood to the fetus. It is also useful for waste removal. The embryo's blood carries carbon dioxide and other waste products to the placenta, where they pass into the mother's blood and are removed by her body. It is useful for protection. The placenta acts as a barrier, preventing many harmful substances and microorganisms from reaching the baby. It is useful for gas exchange. Carbon dioxide from the fetus is exchanged for oxygen from the maternal blood. It is useful for hormonal production. The placenta produces hormones like HCG, progesterone, and estrogen. Let us discuss about an interesting structure that connects the fetus and the placenta is umbilical cord. The umbilical cord is a soft, flexible tube that connects the developing embryo to the placenta. It serves as a life support link between the mother and the baby throughout the pregnancy. Its average length is 50 to 60 centimeters. Its average thickness is 1.5 to 2 centimeters. It connects to the fetus at the navel and to the placenta at the other end. Let us conclude the topic. Learning about the zygote, the embryo, and the embryonic and extra embryonic layers is fundamentally important because it provides the foundation for understanding all of human and animal biology, health, and development. Let us start a biology model lesson on heredity, Mendel laws. First, I will introduce the terms heredity, inheritance, character, trait, and factor or genes. Here, I am using interactive classroom activity idea to introduce the concept.
Acting. Who here looks like their mother or father? What features do you share? Eyes, hair, smile, or habit?
I will start the session with a warm up talk. First, I will ask a simple question to my students. Who here looks like their mother or father? What features do you share? Eyes, hair, smile, or habits? Some of the possible answers may be my eyes are like my mother's. I have my father's curly hair. My grandmother and I both have dimples. Like this, the students will answer to my question. Then, teacher explains these resemblances happen because of heredity, the passing of traits from parents to offspring. Then, I will start family traits observation game by giving some of the instructions. I write a few traits on the board like tongue rolling, dimples, freckles, earlobes attached or free, hair type straight or curly, hand clasp right thumb on top or left, and I will ask each student to observe themselves and note, do I have this trait? Does anyone in my family have it too? They will discuss in groups and prepare a table like this. It contains trait. Do you have it? Who else in your family has it? Some of the students wrote that they have dimples and dimples are present in their mother too. They have no curly hair, but father is having curly hair. They have freckles like their grandma and tongue rolling is like their brother. In this way, students understand the family traits resemblance. After everyone note their answers, discuss what they observed. Then I will introduce the concepts through guided questions and answers. What do you notice about these features? Some traits are similar between parents and children. How do we get these traits from our parents? They are inherited through information passed from parents to offspring. What is this process called? Heredity, passing of genetic information from parents to offspring. What is a character? A character is an observable feature of a particular group of organisms. Characters are inherited as genes by organisms from their parents. Example, hair type, eye color, and height. What is a trait? Traits are the expressions of a character. Example, curly hair, brown eyes, and tall height. What controls these traits inside our body? Genes. Genes are the units of inheritance. A small strand of DNA is called gene. Each trait is maintained by a pair of factors or genes. Mendel called genes as factors. What is inheritance? The way the genetic traits are interpreted. Students can note down the terms and their definitions in their notebooks. Then the teacher encourages the students to read about Gregor Mendel. Mendel was educated in a monastery and went on to study science and mathematics at the University of Vienna. Failure in the examinations for a teaching certificate did not suppress his zeal for scientific quest. He went back to his monastery and started growing peas. Many others had studied the inheritance of traits in peas and other organisms earlier, but Mendel blended his knowledge of science and mathematics and was the first one to keep count of individuals exhibiting a particular trait in each generation. This helped him to arrive at the laws of inheritance. Mendel used a number of contrasting visible characters of garden peas, like they have plant height, tall versus short, flower color, purple versus white, flower position, axial versus terminal, seed shape, round versus wrinkled, seed color, yellow versus green, pod color, green versus yellow, pod shape, inflated versus constricted. He took pea plants with different characteristics, like a tall plant and a short plant, produced progeny by crossing them, and calculated the percentages of tall and short progeny. Students read silently about the Mendel and discuss about him. Then the discussion leads to his experiments. The teacher explains the Mendel laws by using Mendelian coin toss simulation. The materials needed to do this activity are two coins, paper and pencil, chart or table to record the data. Each coin represents one parent's pair of alleles. Here you are observing capital H. fine, and another one is small h. Capital H, that is head, represents dominant allele, that is tallness. Small h, tail, represents recessive allele, that is shortness T. If we toss the two coins, the possible simulations may be capital H, small h, capital H, small h, and small h, capital H. We tossed the two coins together, one from each parent, and recorded the result. The result shows two heads. That means capital T, it indicates tallness. One head and one tail, that is capital T and small t, it also represents tallness. One tail and one head, that also represents capital T, small t, that is also tallness. And two tails, so it represents small t, which is short. So repeat this tossing 16 times to represent 16 offsprings. Count the number of tall and short offsprings. The expected ratio is genotypic ratio is 1 is to 2 is to 1 and phenotypic ratio is 3 is to 1. By understanding the above simulation, we can define the following. Capital H, homozygous dominant. Homozygous dominant refers to a genetic condition where an individual has two identical dominant alleles for a particular trait. Small h is a homozygous recessive. Homozygous recessive refers to a genetic condition where an organism has two identical recessive alleles for a particular trait. Recessive means the allele that is masked or hidden when a dominant allele is present. Therefore, a homozygous recessive individual expresses the recessive trait only because both alleles are recessive. Capital H represents heterozygous. Heterozygous refers to a genetic condition where an organism has two different alleles for a particular trait, one dominant and one recessive. The dominant allele masks the recessive alleles, so the dominant trait is expressed. Distinguishing genotype and phenotype is the first hurdle in genetics. We need to help students to see the difference between the hidden code and visible trait. Genotype, it is the code. This is an organism specific set of alleles for a gene. For example, capital P, capital P, capital P, small p, small p, small p. Phenotype is the observable trait. This is the physical trait that is expressed as a result of the genotype. It is the meal that was cooked. For example, purple flowers, white flowers, black fur, brown hair. Then I will introduce the concept of Mendel laws, the law of dominance, the law of segregation, and the law of independent assortment. The law of dominance shows how a trait can mask another. The law of segregation shows how genes are separated into gametes, and the law of independent assortment shows how tracking of two traits at once. Coming to the Mendel's law of dominance. When an organism has two different alleles, that is versions of a gene for a trait, one allele, the dominant one, is fully expressed and masks the effect of the other, that is recessive allele. The recessive trait only appears if the organism inherits two copies of the recessive alleles. This can be explained by monohybrid cross. When a monohybrid cross is made between pure tall and pure dwarf pea plants, all plants are tending to appear to be tall in first filial generation. You are observing a tall plant here and a dwarf plant here. In F1 generation, we will get all tall plants, which is shown in Punnett square. When these F1 plants are led to undergo self fertilization, both the characters tall and dwarf tend to appear in the second filial generation. In the second filial generation, 75% of the plants are tall and 25% of the plants are dwarf. You can observe these first filial plants. If we self fertilize them, we can get 75% of tall plants and 25% of dwarf plants. This indicates that both the tallness and shortness traits were inherited in the F1 plants, but only tallness trait was expressed. This led Mendel to propose that two copies of factors controlling traits are present in sexually reproducing organisms. These two may be identical, that is homozygous, or may be different, that is heterozygous, depending on the percentage. So, in this, both capital T, and capital T small t are tall plants, while only small t is a short plant. Hence, it is concluded that the dwarf character is dominated by the tall character. This shows the Mendel's law of dominance. Here you are observing another example for a monohybrid cross. If we cross white female guinea pig with black male guinea pig, or black female guinea pig with white male guinea pig, in F1 generation we will get all guinea pigs with black color. If we cross this F1 generation guinea pigs, we will get the guinea pigs in the ratio of 75% and 25%. That is, 3 are black and 1 is white in color. The same monohybrid cross is explained by using shirt buttons. Here I took green buttons as a dominant character and yellow as recessive. In first filial generation, we will observe all green seed bearing plants. In the second generation, you are observing 3 seeds are having green color and 1 seed is having yellow color. If I reverse them, that is green as a recessive and yellow as a dominant, in F1 generation we will get all yellow seeds, and in F2 generation we will get 75% seeds are yellow in color and 25% are green in color. Coming to the Mendel's law of segregation, during the formation of gametes, that is sperm and egg, the two alleles for a trait carried by a parent separate or segregate from each other. As a result, each gamete carries only one allele for a trait. This can be explained by using an example of pea flower color. Here we are observing the parent genotype contains capital P and small p. These are F1 generation plants. They will produce the gametes like capital P and small p. Half the gametes will carry capital P allele and the other half will carry the small p allele. If we self pollinate these first filial generation plants. In F2 generation, we will get the plants having genotypes capital P, capital P, capital P, and small p in the ratio of 1 is to 2 is to 1. We get phenotypes having colors purple and white in the ratio of 3 is to 1. The reappearance of the recessive white flowers in the F2 generation proves that the alleles separated in the F1 generation. Coming to the law of independent assortment. In the inheritance of more than one pair of traits, the factors for each pair of traits are distributed independently to the gametes. This is known as law of independent assortment. Mendel crossed pea plants having round green seeds with a pea plant having wrinkle yellow seeds. All the plants produced in F1 generation have round and yellow seeds, as round shape and yellow color are the dominant characters. On self-pollination of these seeds, plants with round yellow, round green, wrinkled yellow and wrinkled green seeds are formed in F2 generation in the ratio of 9 is to 3 is to 3 is to 1. The above result shows that the factors for each trait remain separate and inherited independently. The appearance of new combinations, round green and wrinkled yellow, that were not present in the original parents, demonstrates that the traits for shape and color were inherited independently. Here you are observing round green seeds and wrinkled yellow seeds. If we cross these two with gametes having capital R, small y and small r capital Y genes, we will get the seeds having dominant character of round yellow. If we self-pollinate these seeds, we will get round yellow, round yellow, round yellow, round yellow, round yellow, round green, round yellow, round green, round yellow, round yellow, wrinkle yellow, wrinkle yellow, round yellow, round green, wrinkle yellow, wrinkle green. Here you are observing round yellow, round green, wrinkled yellow and wrinkle green seeds are in the ratio of 9 is to 3 is to 3 is to 1. After completing the class, students can identify that there are no medium height plants but only tall plants in the F1 generation. Students can learn to calculate the phenotypic and genotypic ratio by using Punnett square or checkerboard showing F2 generation. The students can discuss the pattern of the F2 generation and how do they appear in such pattern. Students can note down the laws of inheritance with examples. At the end of the session, we can ask the factual questions like what contrast characters did Mendel choose for his experiments? Why did he choose the pea plant? What are the genes? What is their role in inheritance of characters? And we can ask open-ended questions like how do genes maintain the traits in organism? Discuss your findings. We can evaluate them by conducting quiz or assignment. Calculate the phenotypic and genotypic ratio of tall and short plants. Calculate the phenotypic and genotypic ratios of yellow and green podded fruits. We can give the homework like observe the ears of all the students in the class, prepare a list of students having free or attached earlobes, and calculate the percentage of students having each. Find out about the earlobes of the parents of each student in the class. Correlate the earlobe types of each student with that of their parents. Based on this evidence, suggest a possible rule for the inheritance of earlobe types.